Hello again. Today we are going to be talking about something a little bit complicated. This will probably be the hardest thing you'll learn in this unit. This is the process of meiosis, how we make sperm and egg cells, known as sex cells. Every other cell in our body, we use mitosis to make. You got to grow hair, use mitosis. You got to grow bones, use mitosis. You got to cut and you got to heal, use mitosis. You need more blood cells, use mitosis. But if you're making sperm or egg cells, you use meiosis. Oh no. And in mitosis, which we've already learned, there was, I picked my apples today. And that was like a lot of stuff to remember. So if in meiosis you've got all these things to memorize again that are even harder, that's going to be really, really bad, right? But here's the trick. Mitosis and meiosis are very similar. If I was being a little bit arrogant, I would almost just say, this isn't exactly accurate, but meiosis is just close to mitosis happening twice, really. There's a couple special things we'll talk about, but it's kind of like it goes, I picked my apples today, picked my apples today. What does that mean? Well, let's freeze this for a second. In mitosis, I picked my apples today. Interphase, prophase, meta, anaphase, and telophase, right? And interphase is planning. This is when everything's in the middle. This is when everything is pulling away. And this is when it tears apart, right? Well, in meiosis, sex cells. What's so special about sex cells? Let me tell you now. They're half. A sex cell is a half cell. Think about it. If a sex cell, if a sperm or an egg was a full cell with all the DNA you needed to live, and it had a full set of chromosomes, and a sperm cell met with an egg cell, and that had a full set of chromosomes, if they joined, you'd have twice as many chromosomes as you needed. That wouldn't be very good. So why we have to use meiosis is we have to take these cells and we have to make them halves. So you'll get a half complete cell, a sperm, and a half complete cell in an egg. And when they join, they'll make a complete cell, which is the new living thing. That's what you are. You're a combination of a sperm and an egg. Each had half from your mother, half from your father. They combine, and that creates you, which is a unique combination of your parents. Kind of cool. So sex cells, and I would also say half. Don't worry, you don't need to write this right now. Just know that mitosis makes 2 cells, right? In the end, meiosis makes 4 half sex cells. Sex cells. Get it? Money? Never mind. And so what happens in meiosis is I picked my apples today, picked my apples today. So just kind of after it does this, it goes back and does another kind of round. That's all you got to know. So if you know interphase, prophase, metaphase, anaphase, telophase, you're probably in really good shape because it's kind of the same stuff. The only place where it gets a little tricky is here. There's a special thing that happens there, and there's a special thing that doesn't happen here. That's all you need to know. So let's get back to the slide and let's talk a little bit. For your assignment today, you're going to have to take notes on what I have done. So if you want to pause the video right now, I'd like you to take this set of notes and put that right here. So right in this section, under the first slide, please take everything I've done. So I picked my apples today versus I picked my apples today. picked my apples today, and I did a little circle here and a circle there, and draw and write down what I've said. 2 cells, 4 half cells, etc. Draw that, get that down there. Pause the video for a second if you need, and I'm going to set up the slides again. So your assignment today is going to be to take notes and send me a picture of those notes. So just like usual, right? So there we go. Couple cool things here. Why is this a great opening slide for something about meiosis? Well, you see that there's actually 4 cells because that's the end of meiosis. When you're making sex cells, you're making 4 cells. The other cool thing I like here is that like some of the pieces look like they've like switched. Like it looks like this blue part from this one actually jumped over to here and the red part jumped over there. And that's really important. That's something called crossing over that we're going to talk about a little bit later. But I love this as an opening slide. So let's just get into it. You've already gotten these notes down off of the board that I wrote. Now, we're here. Meiosis is another name for sexual reproduction, or the making of sex cells, I would say. During sexual reproduction, 2 specialized reproduction cells called gametes fuse to form a one-celled organism. It's called a zygote, and that's called fertilization. So what do you need to know there? You make sex cells from meiosis. They're half cells. When they form, if you get a sperm and an egg and they meet, that is called a zygote. So you can write right here, sperm and egg equal zygote. So once a sperm and an egg cell meet, they become a zygote. Female organisms produce gametes called eggs, and male organisms produce gametes called sperm. And gametes is an important word. Gametes is another word for sex cells, sperm and eggs. So far, so good. Don't panic yet. So if you've made these notes here, you're good. Let's move on to the next slide right here, meiosis overview, and then we'll talk about chromosomes. So a general overview of meiosis. Well, human body cells, every body cell has 46 chromosomes. Hair cells, skin cells, bone cells, blood cells, they all have 46 chromosomes. 23 from our mother, 23 from our father, and they're combined to make us. Every cell has that, right? Each parent contributes 23 chromosomes to every cell in your body is made up of these chromosomes from your parents. This takes place in sex cells, gametes, eggs, and sperms. So what they're saying here is that how do you get these 23 chromosomes? You get them from a sperm or an egg, and those sperm and eggs are sex cells. One cell will become 4 non-identical cells. So what you need to write down here is please just put that, and I'd put an arrow and I'd say because of crossing over. That's important. Now you don't know what crossing over means yet, but basically what we're saying here, and you can draw this here, you're going to get one cell that's going to turn into 4 half cells. All unique. Okay? So let's get a little more into that. Homologous chromosomes. Chromosomes and chromosome numbers. There's not too much to understand here, but you've got to know that, say you have 23 pairs of chromosomes. So you have pair 1. One of those comes from your mom, one of those comes from your dad. Those chromosomes are the same shape and pattern. They could be a long one. And then pair 2, one from your mom or dad, could be a different shape, maybe shaped like an S. Pair 3, maybe a very small chromosome, for example. But every chromosome that you get that's in a pair has a match. One from the mother, one from the father. And matching homologous chromosomes, so what I would do is I would say matching equals matching. So matching chromosomes from one from mom and one from your dad, right? Those matches are going to be the same length, the same centromere position, and they're going to carry genes that control the same traits. So there could be, right here could be a little gene for eye color. This could be for hair color or something a little more complicated than that in the real world, but every matching pair is responsible for certain traits in the offspring. How do you determine whether you're going to have darker hair or lighter hair or certain features? It depends which one of these chromosomes is more dominant. That chromosome will override the other one and show up in the offspring. Kind of cool. One is from the mother, one is from the father. They're a matching set. Now in humans, we would have that. That'd be one matching set. We'd have 23 other matching sets, right? And they'd all look the same, right? And they'd be doing the same kinds of jobs. So you're going to have 23 matching pairs in humans. Let's move on. All right. So meiosis, haploid, and diploid cells. Now what you've got to know is I would just put haploid equals half, diploid equals double. Those are sex cells. Those are regular cells. Okay, so what does that mean? Haploid and diploid cells. An organism produces gametes to maintain the same number of chromosomes from generation to generations. Okay, human gametes contain 23 chromosomes. A cell with n chromosomes is called half. A cell with 2n is called diploid. Okay, wait, let's talk about this. So this is a little confusing. I understand that. So what they're saying, if I can erase this quickly, is a normal cell, a normal body cell would have in its nucleus 23 pairs of chromosomes. And they'd all be matching, right? They'd all, if they had different shapes, right? 23 pairs. One would come from your mother, one would come from your father. And they'd kind of hang out together, right? That is called 2n or diploid. So I'm going to get you to draw this down right away. I'm going to get you to put that right here. Okay. Now you've got to also draw this, right? In a sex cell, once it's gone through meiosis, after meiosis equals, you'd have your sex cell, and inside of it, there would be no pair. There would just be an individual cell. It doesn't have a match, so instead of being 2n twice for partners, it's only 1n. It's got half as many chromosomes because there isn't two, there's only one by itself, right? Let's think about this for a second. So we would call this haploid. So sure, write that down if you need to, but a sex cell does not have matching chromosomes in it anymore. It's got half as many chromosomes as every other kind of cell you've ever known. Now this is where it gets cool. See, the reason they're haploid is because, say this was your mom, and then you had a dad here, all right, or a father, and you would have another cell, and it would have the same division. It would have gone through meiosis, and they would have made their half version. So now, when the sperm and the egg meet, this one will pair with this one. This one will pair with that one. That'll pair with that. That'll pair with that. 23 pairs in humans. I just drew this because it's just a little easier to see. But they would combine, and they turn into a new offspring that's not a sex cell. It's a new thing that's going to grow. It's going to go through mitosis many millions of times and become another person. And they pair it up again. Kind of cool. So you can draw that here. Take your time. Pause the video if you need. And I'll get ready to move on. All right, and let's get the slides back. All right, so meiosis. Now I said there's 2 rounds. I picked my apples today, Well, the first round, I picked my apples today. We call that meiosis 1. And when it gets to the second part, pick my apples today, we call that meiosis part 2. So in meiosis 1 right here, the sexual life cycle of animals involves meiosis. Meiosis produces gametes. You could write there, sex cells. And you can also say that they are 1N. They're half as big. When gametes combine in fertilization, the number of chromosomes is restored. Now take a look at this. This is a great diagram to understand how this all works. So you start off, say you've got a human, and this is the life cycle. Well, they make egg cells and sperm cells, and then they combine. When sperm and eggs combine, you should already know this. The moment they combine, it is called creating a zygote. So this had half the information and half the information, but they combine to a zygote. Now it's got 2N. It's a diploid cell, and that cell will grow through mitosis thousands, millions of times and become a human again. That human will make half or 1N special sex cells through meiosis again. Those will meet up with a partner. Those 2 1N and 1N will combine to make a zygote that is now 2N, and it'll keep creating like that. So here, meiosis. Here, you should actually put mitosis to grow. So let's add that. All right. Awesome. So now let's take a look at meiosis round 1, and then we'll get into round 2 right away. Okay, so now we're talking about meiosis 1. So in meiosis 1, obviously we're going to go through, I picked my apples today, and the special one is going to be metaphase 1, which we'll get to soon. So guess what? First of all, in interphase, nothing new. The chromatin condenses, or you could see the chromosomes. That's what it means. Chromosomes get big and chromosomes replicate. They make copies. Okay, wait. That's exactly the same as mitosis. Good. Less for you to remember. So in interphase, just like in meiosis, the cells condense. They become, the chromosomes condense and they become visible and they make copies. That's all you need to know. So there it is, right? They replicate and they condense. You could say they are visible. All right, let's talk about what happens in prophase. The first round of prophase, I picked. So in prophase, guess what? They're going to pair. So the matching chromosomes that have just copied each other inside of the cell that's going to become 4 half cells later on, they line up, they pair up, right? So the chromosomes pair. Each chromosome consists of 2 chromatids. The nuclear membrane breaks down and the spindles start to form. Wow, that sounds very familiar. It's the exact same. Pretty cool. So in prophase, once again, the pairs line up, they get together. Spindle fibers start to form, the nuclear membrane breaks down. Easy stuff. It's the exact same as mitosis so far. Now, coming up next is the trickiest part. Right here. This is the coolest part. So, on the last slide, you could see here I said metaphase is special. Well, it's kind of, both of those are special. But this is really important here. So I'm going to write very special. This is important, folks. In prophase 1, crossing over happens. You see, because let's go and think about this for a second. All right, so let's pause the screen. Let's take a look. If this was you, say you were making a sperm cell or an egg cell. All right, so you're going to make some sperm cells and some egg cells. All right. Well, guess what? If you went like this was your cell to start with before it divides, and you had in your nucleus, you had all your pairs of chromosomes. So you had 23 pairs because you're human. I've only drawn 4. Now, if there wasn't this thing right here called crossing over, which is very important, exchange of genetic information. If there wasn't crossing over, which I'll explain what crossing over is before they split up and say goodbye in the next phases, in anaphase and telophase later, they actually switch parts. So what happens, let's zoom in on one of these. If you had this chromosome here and its partner, what would happen is this part might switch with this one. And this part might switch there. And they're actually going to change pieces so that they're not exact copies. They're going to take a little bit of each. Why do they do that? Well, let's think about this from genetics. The more diverse a species is, the better chance it has to survive things like plagues and all kinds of environmental changes and things like that. So diversity as a species in humans actually makes us much stronger. So, where did this chromosome come from in you? And this one, and this one, and this one. I'm using the left side as an example, but one chromosome always comes just from your dad, and one chromosome always came from your mom. So if your chromosomes just split like this, that would be bad because you'd either be giving to your offspring an exact copy of your mom or an exact copy of your dad, depending on which one ripped where when you were making sperm and egg cells. That's not very good. That's not very diverse. So before these chromosomes from your mom and dad split up, they actually exchange parts to make a new combination of genetics that's going to make the species more diverse and more resilient. So instead of just splitting up the chromosomes before they're going to make these sperm and egg cells, saying, oh, this side, all the sperm and egg cells will be made from your mom's genetic material, and all the sperm that comes to this side, or eggs that are made on this side, will be made of your dad's genetic material. That's no good. What we want to do is we want to mix parts up and create brand new genetic combinations in every sperm and egg cell through something called crossing over. So we can go back. All right. Ooh, that's a little chaos. All right. So crossing over produces an exchange of genetic information. In crossing over, chromosomal segments are exchanged between a pair of homologous chromosomes. So that just means that matching chromosomes from your mother and your dad change parts before they split up. All right. That is probably the most important thing. So I would say creates diversity. Now I'm not talking like, oh, diversity. I mean like genetic diversity, new combinations. Okay, let's be clear on that. Now, in metaphase 1, well, guess what happens in metaphase, folks? What happens in every metaphase you've heard of so far? Metaphase means middle. What happens here? Let's see. Chromosomes and centromeres attached to spindle fibers, homologous chromosomes line up at the equator. I love this diagram too because you can see the parts crossing over in it. Now in your copy you can't, but up here you can. You can see that like these little chromosomes that are about to rip away forever, they've actually exchanged information. Kind of cool. So metaphase means everything lines up at the middle. Guess what? That's just like regular metaphase. Guess what's after metaphase? I picked my apples. Anaphase. Anaphase means away. Are we correct? Let's see what happens in anaphase 1. Homologous chromosomes separate and move to opposite poles. That sounds familiar, doesn't it? Because it's the same thing again. Anaphase is anaphase. Okay. So then we get to telophase. What happens in telophase? It's the same old, same old. You've actually ended up with 2 cells. So the spindles break down, chromosomes uncoil and form 2 nuclei, and the cell divides. Okay, so far so good. I picked my apples today. The most important thing was in prophase / metaphase, there was crossing over to create mixtures of your mom and dad's genetic information before they split up to create sex cells. So now, you've now ended up with 2 cells. You've got 2 cells. Now they're going to divide one more time to become 4 half cells. Now this is the next part that's very important. Notice how there's no interphase. But what happens in interphase? Interphase is when the chromosomes double. But we don't want doubling the second time around. Because if we doubled again, we'd have too much DNA. Let's pause and take a look at this, and let's think about this for a second. So, what we'll write here is we will write no doubling in interphase. There is no interphase here. So let's take the video down for a second. Let me clear the board. Think about what I'm saying here. We want, on one end, right? We start here with the cell, and in its nucleus, we'll have I'm just using 4 because it's easier to draw, but you would have something like this. And in the end, what we want, let's be very careful here. In the end, what we want is we want 4 cells that each have just half of the information. Notice how these cells don't have matching pairs like this one did, because these have to actually meet up. If this was sperm cells, they'd have to meet up with an egg cell and become matching partners, one from the mom, one from the dad, right? So if all of a sudden a sperm came here, this was an egg, and it had, from another parent, had this, and this, and this, and this, then all of a sudden they join up, they all match, and that becomes an offspring. But we want this in the end. So what we did the first time is we did double. There was an interphase in the first version, right? Remember, it goes, I picked my apples today, picked my apples today. There is no interphase. And remember, crossing over happens here. Those are the 2 most important things to learn about meiosis. So we've made 2 cells, but we copied because we had an interphase the first time. So we've actually just ended up with basically the same thing the first time, right? So, okay, we doubled the genetic information, but guess what, guys? If we doubled it again, right? So this is this whole process. This is called meiosis 1. And now we have to go through meiosis 2. If we had another interphase here and they doubled again, we'd end up with full cells again, 4 full cells. We don't want to do that. We want to end up with 4 half cells. So we don't do interphase the second time. We just take these now, and all we're going to do is just rip them apart. Take these two, rip them apart. Take these, rip them apart, and we're just going to split these up. We're going to split every one of these, right, and send them to half cells. They're not going to have a matching partner anymore because the matching partner is going to come from the mate, from the other parent, and that'll match to create a new combination. So that's why we don't have an interphase. We don't double the second time. In meiosis 2, we do not double genetic information. In meiosis 1, we double it. We do crossing over, which is that special dance to make sure you're giving a good combination of your mom and your dad's DNA to your cells. They create copies, and then we rip those apart and we make half cells. No duplication. All right, that's the most important stuff, so let's see what we're at here. So, in prophase 2, a second set of phases begin, the spindle apparatus forms, and guess what? They're going to grab them, they're going to bring them to the middle, they're going to rip them apart, tear, and you're going to end up with 4 cells. Picked my apples today. So prophase, spindle fibers come out, they're prepping, they're getting ready, just like the previous prophase, remember? All right. Guess what happens in metaphase? Look at that. Middle phase. Meta means middle. So metaphase, a haploid number of chromosomes line up at the equator. In anaphase, the sister chromatids are pulled apart by the spindle fibers and moved to opposite poles. That's exactly what normally happens in anaphase, right? Except now you can see that they're not partnered up anymore. They're only they don't have a partner. This chromosome and this chromosome are going to be separated. They're not going to have their matching set anymore. Which brings us to telophase 2, right here. And in telophase 2, the chromosomes reach the poles and the nuclear membrane reform. So this would now be 4 half sperm or egg cells. And in meiosis 2, cytokinesis results in 4 haploid cells. This is important. 4 haploid cells. Genetically unique. Now, the importance of meiosis. Meiosis consists of 2 sets of divisions. Meiosis 1, I picked my apples today, and meiosis 2, picked my apples today. Produces 4 haploid cells that are not identical, results in genetic variation, as we've talked about. There's crossing over and there's ripping of it apart. The odds of you existing are 1 in 4 trillion or something. The odds that your DNA was mixed and taken from your mother, the odds that your mother and father met, that how when their DNA crossed over, which one of those 4 cells met with which egg cell or sperm cell randomly, very long shot that you're existing right now. Kind of cool, very genetically diverse. And meiosis provides variation. Depending on how the chromosomes line up at the equator, 4 gametes with 4 different combinations of chromosomes can result. Genetic variation is also produced during crossing over and during fertilization when gametes randomly combine, because men produce millions of sperm cells. So there's already 1 in a million that you exist, right? 1 sperm cell. And which egg cell is ovulating at that time is completely random too. And the combination that made that 1 sperm cell and the combination of crossing over that made that 1 egg cell that met at that one time, 1 in trillions. Now, this last slide, not that important. So what I want you to do is, I know this was a long lesson, but what you've got to remember, let me mute this, is that you start with 1 cell, you go through meiosis 1. I picked my apples today. You should know what a metaphase, anaphase, and telophase are because it's the exact same as mitosis. and you've made a copy. The second time, you skip interphase and you just start ripping them apart and you end up with 4 half cells. You've got to remember that there is no interphase 2. You've got to remember that in prophase 1, crossing over happens, which is exchanging of genetic material between matching pairs of chromosomes. So I hope you understand what meiosis is. Remember, meiosis is this process is only used to make sex cells. Every other, which is sperm or eggs, we call those gametes. They're haploid. They're 1N as opposed to 2N cells, which is every other cell you've ever heard of. So 2N cells are diploid. That's body cells, hair cells, blood cells, bone cells, every cell you can think of. But sperm and egg cells use meiosis. They go through meiosis 1, they go through meiosis 2, and you end up with 4 half cells. So that's one partner. This is like the mom made 4 half cells, and then the father would make 4 half cells. 1 half cell would randomly meet and create a new form of life. Pretty cool. So I hope you've understood that. This is the hardest thing you're going to have to learn. So don't be too upset if you don't get it. But ask questions about this because this is an important concept and we're going to be talking about this a lot more and it's going to be a big component of your test. So if you need clarification on meiosis, please ask. This is the hardest thing you're going to learn this unit. So good luck. And your assignment was just to make all the notes. And please, I'm sorry, at the end, right here, draw what I have drawn here, okay? Take this, draw that, put it on there. Take pictures of your notes and send them to me. If they're not attaching in Google Classroom, just email me, put the lesson number in there. We're good to go. Please send them, I'll mark them, and we are going to be moving on to some worksheets and some micro viewers about meiosis now. It's the last thing we do before the reviews. Good luck, folks.